Please welcome to the stage President and CEO, BioCrossroads and Central Indiana Corporate Partnership, David Johnson.
Good morning. It's like a summer day.
It's the first time there's ever been music to an introduction for me, but thank you very much. Good morning and welcome to our 11th annual Indiana Life Sciences Summit. It's a pretty amazing thing that we've been doing this for 11 years, and it pretty much tracks the progress of BioCrossroads and the BioCrossroads initiative that's been created here in Indiana. The program is much as you've seen before, but the content today is all new. And it's just a really exciting event. I say this every year, but it's true every year, that the content of this just keeps getting better and better. We have a terrific keynote speaker and three tremendous panel discussions on topics that are of great interest to most of you here, if not all of you here, each one of them. And we have some other special events that we're going to have during the day here as well. So I want to thank our colleagues at AlpInvest, who with BioCrossroads co-sponsor this event, as well as our many other wonderful co-sponsors, including the Cook Group, Eli Lilly and Company, Roche Diagnostics, Faegre Baker Daniels, and Miles Printing. People will be coming in and out of the conference through the day, but we have record attendance and registration for this event, and we are committed to do justice to the interest that all of you are showing in it. And to make that really work and make it happen the way it should, to do justice to the program itself, it's a particular pleasure of mine to be able to introduce you once again to our able, seasoned, battle-tested master of ceremonies who will actually make this whole ambitious agenda work. So please join me in welcoming our friend and colleague, our founding BioCrossroads and Biosciences Research Institute board member, Dan Peterson, Vice President of Industry and Government Affairs for Cook Group. Thank you, Dan.
Thank you,
Take
David.
it away.
Thank you. Thank you all very much. It's really great to be here. 11 years, that's amazing, a decade of these things. Thanks to, luckily for all of you, I have not been doing this role for 11 years, and if I keep doing it, we'll see. But it's a great pleasure, great honor to be here. Thank you to BioCrossroads. Thank you to AlpInvest, to Lilly, to all of our other corporate sponsors. We look forward to a fabulous day. I think, as David said, you're going to hear from some excellent speakers on critical topics to our industries, and hopefully share some good interchange with lots of other conference attendees throughout the day as well. It's been a busy year since we last met in this very room, I believe. Certainly the IBRI, the Indiana Bioscience Research Institute, continues to gain significant momentum, and I believe that that organization, that effort, that collaborative is very emblematic of why Indiana has been so strong and is so strong in life sciences, but why I think our opportunities are just on the cusp of really growing dramatically. And so to thinking about the Institute, thinking about all these efforts around collaboration in this industry brings together the very, very strong corporations, universities, healthcare organizations, inventors, and all for the good of patients and opportunities for patients to improve their lives. So it's a noble and worthy endeavor that we're all involved with, and I, for one, am very, very proud and excited to be part of it. We continue to see evidence that we're doing the right things. We can always do better. Indiana continues to be, now for the last couple of years, the second highest exporter of life sciences products in the United States, second only to California. And absolutely. And one I didn't know until today, for the first time, we hit $100 million in venture capital for startups. We hit that mark in August, so another significant milestone. Let's see that number keep going up. Today you're going to hear from a really prominent, excellent speaker on the national healthcare scene and the politics of healthcare innovation. We're going to discuss the IBRI, we're going to talk about what the future holds for the capital markets, and we're going to recognize a visionary within our life sciences world. A couple of pieces of protocol. When you ask questions from the audience, there should still be, or there are microphones posted in the hall or in the aisleways, if you will. You can tweet your questions using one of the hashtags listed on the sheets in front of you and on the nope, not right now on the screen, but on the screen at some point. You can also tweet from the conference using those same hashtags. So, let's get the day started. I'm very honored to introduce our keynote speaker as my next task. This morning, our keynote speaker is Dr. Christopher Austin, and he's the director of the National Center for Advancing Translational Sciences at the National Institute of Health, a very critical organization in our world for sure. Dr. Austin was appointed to this position by NIH Director Francis Collins in 2012. His mission at NCATS is to catalyze the generation of innovative methods and technologies that will enhance the development, testing, and implementation of diagnostics and therapeutics across a wide range of human diseases and conditions. Dr. Austin is applying his experience across the spectrum of translational research to identify commonalities among diseases and implement a system-wide approach to accelerating the translational sciences process, thus speeding delivery of interventions that can really improve human health. Dr. Austin came to NIH from industry, actually came from Merck in 2002. He began his career at NIH as the senior advisor to the director for translational research at the Human Genome Research Institute, where he initiated the Knockout Mouse Project and the Molecular Libraries Roadmap Initiative. I understand the second one, not quite sure what the first one is, Knockout Mouse. Either a large mouse that needed to be taken care of or a really good-looking mouse model, I guess, right? So he'll explain, I'm hoping, certainly. Dr. Austin earned an A.B. summa cum laude in biology from Princeton University and an M.D. from Harvard. His group is the one that actually implements and awards the Clinical Translational Science Awards and has awarded 2 of those, in fact, to Indiana University School of Medicine and the consortia of universities that are a part of that important initiative here in Indiana. We're very excited and eager to hear from him and look forward to his talk. Little known fact, Dr. Austin is an accomplished opera singer, and therefore we are asking him to deliver his talk a cappella and sing for his breakfast. So without further ado, please help me welcoming Dr. Christopher Austin.
Wow, I do like that jazzy music. That's really... welcome to my talk. I'm not going to do this in recitative, although I could. Be kind of fun, actually. What I'm going to do today is give you a sense of what NCATS is trying to accomplish. We're the newest member of the research ecosystem at NIH. There's 27 institutes at NIH and centers. We're a little over 2 years old, so I sometimes say we're in the terrible twos. We have a lot of potential, but we're still growing, still finding our way, make a mess now and then, like a good 2-year-old. But what we're doing is quite different, and I think a lot of what we're trying to do, I think, will resonate with what you're trying to do in Indiana. So, okay, so I stole a few Francis Collins slides to start out here, it's just to orient everybody. So NIH, as you probably all know, is the steward of medical and behavioral research in the United States. It has a dual mission, which is science in pursuit of fundamental knowledge about how systems work as a basis of applying that knowledge to improve the burden of illness and disability. Just to start on a sobering note, because when I was asked to do this, one of the themes apparently is at our place, as it is in yours, is managing in a constrained budget environment. I suppose we're always in a constrained budget environment one way or another, but this really is an interesting time at the NIH. This is actually the funding of the NIH. If you look at the green line, it's a historical increase over the last number of decades. It's a 3.7% increase annually. And you'll notice during the doubling, that went off that historical growth rate in the positive. But since then, the last 10 years, actually has been what's sometimes called the undoubling. so that the NIH budget, now another way to look at it, is in real dollars, which is the inflation-adjusted dollars, which is the purple line here. We have about the same purchasing power as we did in 2001. From its peak, the purchasing power of NIH, the NIH budget's down about 20 to 25% since its peak at the end of the doubling. This has really put remarkable strains on the system, and it's particularly painful for those of us who work in translational science, because I think we're well aware that the opportunities to really do good are phenomenally greater than they were when I was in training 30 years ago. And the other thing to just kind of keep in mind, if you haven't seen this, is other countries are continuing to increase their scientific research and development spending. Of course, China being a major one, with the U.S. being negative over the last few years. Of course, what this isn't showing you is the absolute numbers. NIH is still, U.S. is still quite far ahead in absolute numbers, and will be for a little while to come. But of course, as we all know, those absolute numbers and the positions of various nations can change actually quite rapidly, and so a number of us are really concerned about this trend. So that's the bad news. Thought I'd get that over with. But I did want to sort of put this in context of what we're dealing with in Washington. This is another Francis Collins slide talking about some of the major opportunities as we see them at NIH in biomedical research. It's a huge number of new ways of unraveling new basic mechanisms through basic research, focusing on big data and technology, translation, which is what I'm going to talk to you about, and then the workforce, which is an issue, again, that all of us really worry about in this era of constrained resources. So I'm just going to mention one thing about on an NIH-wide level, and then I'm going to dive into NCATS a bit, about translating discovery to health. You may have seen a few months ago that Francis announced this really interesting partnership with a number of pharmaceutical companies and nonprofits called the Accelerating Medicines Partnership. It's really, at its basis, it's a target validation initiative, really. But I think somebody decided that target validation didn't have the kind of press-friendly sort of connotation that accelerated medicines did. But if you want to know what the organizations are really doing, they're doing target validation and trying to identify new targets for a number of really important diseases. And these are the diseases. They're Alzheimer's disease, type 2 diabetes, and a variety of autoimmune diseases, among which are lupus and RA. And these are being run out of the three institutes at NIH that are responsible for these diseases: the National Institute on Aging, National Institute of Diabetes and Digestive and Kidney Diseases, and the National Institute of Arthritis and Musculoskeletal Diseases, respectively. And the costs are about equally borne. It's a several $100 million initiative, and they're equally borne between the public and private sector partners. So it's a pretty exciting initiative, which I think really gets at the kind of public-private partnerships that you're trying to put together here in the state of Indiana, which is why I mentioned it. Okay, so on to translation. One of the things that really struck me when I became director of NCATS about 2 years ago is that we are at a disadvantage to our brother and sister institutes because of the word translation. When I first started going down to the Hill to talk to Congress people about what we do, the typical comment was, okay, so we had the folks from the National Cancer Institute down here. I know what they do. I know what cancer is. We had the folks from the National Eye Institute down here, and I really don't know how the eye works, but I've got one. Most congressmen actually even have two. And so you guys are from the National Center for Advancing Translational Sciences. So you guys do what, linguistics? Is that what you do? And right? Because that's the term that's used. For those of you who are scientists, I've actually had scientists say, you have an entire center at NIH that's focused on how mRNA gets made into protein? That's amazing. That's a really specific center, because that's the biomedical term of the word translation, molecular biology, anyway. So how do we define translation? And we really define it broadly. It's the process by which an observation is made, the process by which an observation that is made in the lab or the clinic, or it could be made in the community, where a light bulb goes off and somebody says, this is a really interesting insight that may lead to an intervention that could improve human health. That process from that, when the light bulb goes off, to creating the intervention, to showing that it can be useful to improving human health, to showing that it is useful in a public health setting for actually improving human health. That whole process, multifaceted process, is what we call translation. And it applies to diagnostics, therapeutics, devices, behavioral interventions, procedures, et cetera. But you'll notice that NCATS is not the National Center for Translation. Virtually every NIH institute, all 27 of them, do translation given this definition. We are the National Center for Advancing Translational Sciences, and there's two differences. One, it's often been pointed out, and I like this distinction, we are the only NIH institute or center that has a verb in the name. And we like that. We are rate of change people. We are people who are really dissatisfied with the current state of affairs. And so we are constantly asking, how can we do better in a general way, lift all boats so that the whole translational process works better, faster, cheaper. And secondly, we're about translational science. So what the heck is translational science? Well, it's the field of investigation that seeks to understand what the general principles are underlying each step of the translational process, whether we're talking about medicinal chemistry or toxicology or pharmacology or formulation or clinical trial design or what have you. There are general principles that frankly are not well understood, and in my opinion, that's the reason why we have such a tremendously high failure rate in this process. It's a remarkably empirical process, those of us who do this. And whenever you have a process that has so many possible degrees of freedom and you have a trial and error approach, the likelihood that you're going to have an error is much higher than you're going to have the likelihood that you're going to have a success. And that, of course, is the history that all of us have dealt with. And so the only way to fix this is not to throw more shots on goal, not to have more shots on goal. When I was at Merck, actually, we were going through the shots on goal era, and a lot of you probably remember that. Didn't work. The numbers are just not with you. You have to understand what you're doing first. And there's really never been an organization in the world that's been tasked with doing this. And I'll get down to what we're actually talking about in a second. Okay, so sometimes I'm asked, well, why is this so important now? Well, I think it really is important, particularly important now, because we live in what is really a painfully paradoxical time, where those of us who work in science and technology get to deal with every day with fantastic technologies, which are just things which we never could have imagined, 20, 30 years ago. The fact that I often tease Francis by saying that, the whole Human Genome Project, you can do the Human Genome Project now in a robot the size of a toaster. Right, so you could fit Francis's entire career into a toaster, and that's a great thing. It's just like being able to take a mainframe computer and put it in an iPhone. But those of us who work on interventions to improve human health and patients who have diseases who are waiting know that health has in no way kept up with these epical changes in science and technology, because we are just not very good at understanding how to transition from what's on the left side, fundamental discoveries, to what's on the right side. And NCATS's goal over the next 20, 30 years is going to be to prevent this kind of IOM report that was written a few years ago from being written. And if you haven't read this, if you want to be depressed, probably have enough reasons to be depressed, but if you don't, sort of in a manic phase and you want to bring yourself down a little bit, you might want to read that. It's called, it's an IOM report called Shorter Lives, Poorer Health. And it goes on in great detail about how unnecessarily unwell the American public is. And one of the effects of this, and you see this, note this at the bottom, is that funders in the public sector and the private sector have either gotten impatient with us or have lost patience with us altogether. If you look at the flight of capital from the life science markets in the last decade, I don't need to tell you, this is a huge issue. And it's an issue with our relationships with Congress as well, and the American public. So I think this is a fixable problem, but really, I think we really need to focus on it. Okay, so one thing we're often giving you, and again, I'm gonna depress you, and then I'm gonna pull you back from the abyss. So just hang on, it gets better. We're often compared to the microprocessor industry, and you all know this, that this is Moore's law, is the linear increase in the number of transistors you can put on a microprocessor over the last 40 years. The reason why you have an iPhone that's the size it is with the power that it has, and the cost has come down in a similarly linear fashion. But we in drug development, of course, have Eroom's law, which is Moore's law spelled backwards. If you haven't read this paper, you really should. It is terrifying. And it's terrifying because what this shows is dramatically negative productivity growth in the pharmaceutical development business over the last 60 years. Since 1950, the number of drugs approved by the FDA per $1 billion spent has gone down by half every 9 years since 1950. Now, if you think about the definition of productivity growth, right, more widgets, same amount of money, this is dramatically negative productivity growth. This is why pharma is having so much trouble. Nobody can make a business out of this. And I would argue that a lot of the reason for this, not the only reason, but a lot of the reason is we don't understand the underlying principles of what we're doing. We're approaching this as an empirical process, and we have to understand this process better. The reason I find this particularly terrifying is two reasons. One, NCATS was formed in part to reverse this curve, and secondly, if you ask when this line goes to zero, which I did a few years ago, extrapolated out, goes to zero in 2040. So unless something changes formally, there will be no more drugs after 2040. Or in other words, the cost of a new drug will be infinity, which is pretty much the same thing. So we have got to fix this problem. And think about the absolutely incredible changes that have happened since 1950, and none of them have changed the slope of this curve. So, you depressed enough? Okay, no, so. The good news. We know a lot more than we did before. So this is just one example of it. Disorders with a known molecular basis. Before the Genome Project was done, very small number, genes for cystic fibrosis, Huntington's disease, sickle cell anemia, those kinds of things. But with the deposition of large amounts of sequence data in public databases in the mid-90s, the number of Mendelian disorders, the genetic basis of which is understood, has really exploded to over 5,000 now. Of course, and so we have an ability, at least a context, an understanding on the basis of which to intervene, which we never had before. The bad news is, of course, for patients, is there's only about 250 of these that have any treatment currently. Some of this is simply a time lag. If you take the normal 15 years that it takes to develop a drug, some of this is just a time lag. But we are never going to solve this problem in a brute force way. If you do these numbers, there's just too many diseases, and the unit cost is simply too high. Okay, so what I learned as a neurologist, I'm a neurologist and a geneticist by training, and those of you who know neurologists and geneticists will know that they have one thing in common, which is that they are really good at diagnosing things, can tell you in great detail what's the matter with you, but are completely useless at actually doing something about what you have. It's a little apocryphal, but it's basically true, and I can say that because I'm both of them. And so what I learned was that as a neurologist, I could diagnose this, and even the orthopedic surgeons in the room could probably tell me what this is. That's a brain, by the way, and the big black thing, that's a stroke for you orthopods in the room. Sorry. And neurologists are great at this. Oh, Mrs. Jones, you have a stroke. It's right in this region of the brain, and of course she looks at me with a blank stare to say, and I say, aren't you pleased? I told you what you have. And she said, no, I'm not pleased. I want you to fix it. No problem. So geneticists are the same way. Okay, so this is the original paper diagnosing sickle cell disease, 1949, Linus Pauling, the molecular defect in sickle hemoglobin. 65 years later, there is still no drug directed to the molecular defect in sickle cell. Right? Diagnosis great, can't treat it. So most patients realize, although most doctors or most scientists probably don't, that this and this is not equal to this. And what patients want is this. And when you think about the problem in translation is, at least in the academic world, we don't really train people to fix things for the most part. In the science world, we train them to understand how things work, and when things go wrong, why do they go wrong? But we don't train them to fix it. And it's actually quite a different skill set. So you can think about translation as the how to fix it. Sort of skills. Doctors, of course, are trained how to in this, but of course it's a very empirical sort of a black magic sort of approach. It's not really a scientific approach. And again, as a physician, I can say that it really is true. So into this maelstrom comes NCATS. Okay, so what is NCATS? And you heard this is our mission. I just want to pick up a couple of words here. We're a catalyst. And we view ourselves as a catalyst. We are 2% of the NIH budget, so we have to be like a good catalyst, present in small amounts, but are very productive. How does a catalyst work? A catalyst does not do anything by itself. A catalyst is obligatorily collaborative, and we are obligatorily collaborative. We've designed the center that way to not be able to deliver on our programs, on what we have to do by ourselves. So every project we do is a collaboration. So biochemically what a catalyst does is it brings reagents together, puts them in close proximity, and allows them to produce a product that they wouldn't otherwise. And that's exactly what NCATS does. Different from our brother and sister institutes. So what are we catalytic of? New ways of doing things, new paradigms, technologies, data that increase the speed and efficiency with which interventions are developed and shown to be useful in improving human health. And the last phrase just says that we're disease agnostic. We have all 7,000 diseases which affect the human family that we're interested in. And we are fundamentally, our approach is fundamentally different from the rest of NIH. And NIH is organized as 27 institutes, and they're generally organized the way doctors are, right? So there's heart doctors and cancer doctors and eye doctors and brain doctors. And so at NIH, there's a heart institute and a cancer institute and an eye institute and a brain, various skin institute, et cetera. But we all know that actually they're all connected. And so NCATS is an integrative institute. We think not about what is different about organs and diseases. We think about what is common to them, because we're big believers that the knee bone is connected to the leg bone, and if you approach the problem this way, you will be able to make advances which you otherwise couldn't. And we have to be aware that the current science funding system and promotion and tenure system frequently mitigates against these kinds of leaps from being made, which Mother Nature wants us to make, but the system we have created prevents it. And I've had this problem in my own career, and I'm sure a lot of you probably have. Okay, so I said that NIH is different, and it really is. NIH, this is sort of I love this slide because it was actually made by Francis's office. This is as an org chart of the National Institutes of Health, I like to think about it as an aerial view of the silos that make up NIH, because each of these little billiard balls is a different institute. And NCATS really is viewed as a horse of a different color here, but it's also viewed as a connector. An adapter, an integrator. But we're also an integrator across the larger biomedical research ecosystem. Certainly every institute at NIH has these kinds of collaborations, but because of the place that we operate in, for instance, we do no basic research. NCATS does no basic research. We start at target validation, if you want to use that vernacular. We rely on again, the 26 institutes that we work with to do the fundamental research. We absolutely rely on that as our seed corn. But we are sort of right-shifted, if you want to think about it that way, in the development spectrum. So we have much more robust and regular interactions with disease advocacy groups, nonprofits, FDA, other regulatory agencies, biotech, pharma, et cetera. Now, let me just say something culturally here, because this is a big deal, particularly for academia. It's a big deal for pharma too, although they do a better job with this. And it's a major limitation in academia playing a big role in this. And this is something that you're dealing with here in Indiana and trying to create teams to do this. So what is a team? A team is something that requires top performers in a variety of different, with a variety of different expertise to work together to a common goal. That's basically what a team is. And there is this canard out there in science, which a lot of you probably aware of, that I was certainly brought up with, which is that individual effort is the only effort which has any validity in science. And that team science is not science because it devalues the contribution of the individual. To which I say, give me a break. That is just hogwash. It's like saying that LeBron James is not a good basketball player because he plays on a team. It's a different sport. It depends on what the sport is that you're playing. And conventional science in academia, how I was trained, this was my exemplar. Does anybody know who this is? Yeah, it's got to be Fleming, right? Guy, black and white photograph, crud on a petri dish, that sort of wistful view in his eye. I was growing up with this. We're solitary investigator scientists working by candlelight at 3 in the morning, eureka moment. And it's wonderful. I've had those events myself. It's incredible. Dopamine rush, better than cocaine. I don't know if it's better than cocaine. Never done cocaine. I don't know what's better than cocaine. I hear it's better than cocaine. But it is a wonderful thing that drives curiosity-driven fundamental research, and this is incredibly important. But this is science as golf. It is a solitary exercise, sometimes with a caddy, otherwise known as a lab head. But that is not translation. Translation is football, and at this point, I have to point out that I'm a Baltimore boy. So to me, and I just got to get this off my chest because I'm in Indianapolis. So I grew up with this. Now this is a Colt. This is not an Indianapolis cult. This is a Baltimore cult named Johnny Unitas. And this is not Andrew Luck. And we're still mad in Baltimore. Okay, just had to get that off my chest. So what happens in football is that you notice that there's a quarterback. For those of you, and I was just talking to Annalisa about this, so all you Europeans who think football is a different sport, bear with me here. Actually, I have a German woman who now runs our whole clinical part, and she says, in the last 5 months since I've worked for you, I have learned more about football than anything else, because I tend to talk in these analogies. But it's actually very useful. Okay, so what happens? So quarterback is handing off to the running back. The running back's going to try to run around and score a goal, right? And he's never going to score a goal because he's got a really strong defense. And look at all those guys in the white, right? Think about those as toxicity, lack of efficacy, pharmacology, formulation, clinical trial design, really strong defense, right? So the only way that the guy is going to score is you see I think it's number 7, what is he, I can't see the white guy to the left. He's the defensive lineman. He's going to try to take out the running back, right? And the only reason why he's going to score is that number 72, that offensive lineman, is going to throw his body in front of the defenseman, risk life and limb to help his teammate score. And the reason he does that, because if the teammate scores, number 72 gets credit too, right? Now, this is in academia. You score, you get a paper. There's only 2 people who matter, first author and last author. The first author is the guy who actually makes the score. That's the running back. The last author is the guy who's calling the plays. That's the lab head, right? That's the quarterback. Everybody else in between, all the middle authors in the paper, no credit, don't get tenure. And then you ask why people don't do translation well in academic settings. Well, duh, because we're not incentivizing them to it. We're disincentivizing them to do this. So I talk to trainees all the time who want to do this, but they're saying to me that you've got to change, you, me, us, have to change the system to allow them to do what they want to do. Okay, so translation. For those of you who don't use this vernacular, I do find it useful. So what NCATS does is not only in the T1 world, that is going from target to proof of principle in human, sort of phase 2 trial, if you want to use that terminology, but also T2, which would go from a phase 2 trial to approval, FDA approval, or showing to be useful in a large clinical trial, but still this hothouse environment of a clinical trial. T3 going to routine medical practice, and T4 being to show that this intervention actually does improve human health. And so NCATS works across this spectrum. And sometimes people ask me, well, what are the problems you guys work on? So I understand these problems, I understand what you guys are trying to do, but you go in on Monday morning, what do you actually do? What's the problem list, right? So frequently I say, and those of you physicians will know this term, physicians always talk about problem lists, that the patient has, hypertension, diabetes, dyslipidemia, had a stroke, MI, ingrown toenail, et cetera. That's a problem list. Well, NCATS's patient is the process that is sick, and this is the problem list. These are all the problems that affect any translational process. And you will notice that you'll notice that there's no disease names in this list. They all fit what economists would call the tragedy of the commons, which are everybody's problem in general, but nobody's problem in particular, so nobody works on them. And up until NCATS was formed, there was no institution in the world whose day job it was to solve these problems. Right? So at NIH, so the predictive toxicology problem, whose job is that? Is that the Cancer Institute or the Neurological Institute or the Eye Institute? Well, so the answer is nobody's. Is it Merck's job or Pfizer's job or AstraZeneca's job? Well, it's nobody's job, right? It is our job. And similarly, all the others as well. But we're acutely aware that even if we got the science right, there are many, many operational problems that would still prevent many, if not most, translational projects from moving forward because the culture isn't right. So this is the cultures of data transparency and data release is a big issue in the clinical world, but it's a big issue at NIH in the preclinical world as well. How to handle IP, integration of project management into the academic setting, which project management is motherhood and apple pie in a pharma. It is anathema to an academic scientist. I can tell you, I have been the words that have been used for project management cannot be published in a newspaper in the academic world. They're terrified of it. and for a reason which is just its unfamiliarity. So integration of project management, credit for team science. We talked about this before. Credit for health improvements. What's interesting, and Anantha Shekhar and I have talked about this a lot. What's interesting is that in many academic settings, you can get credit, you can get tenure for having a paper in a journal on a signal transduction pathway in yeast, which is really important. Don't get me wrong. But if you make a major health improvement in a health system where thousands of patients get better because of the intervention you do, you won't get a high impact publication, you won't get tenure. There's something wrong with a system that does that. So education and training in the area of translation. I had to go to pharma to learn the black art of that all of you do. And so how do we have academics learn this without having to go to the dark side to do this? And then novel collaborative structure. So all of these are areas of innovation that we focus on just as much as we focus on innovation in science. Okay, so what are the scientific initiatives? We basically split them into clinical initiatives, preclinical initiatives, and re-engineering the translational process. And I'm just going to run through a couple of these. The biggest program, and you heard this mentioned already, is the CTSA program. It's a national consortium of medical research institutions that work together to improve the way the clinical piece of the translational process is conducted nationwide, and provide training, innovative training for clinical translational researchers. And this is what the map looks like now. It's quite a remarkable collection of organizations. In case you got lost and came to the wrong meeting, you are here. And the person who runs this is Anantha Shekhar. Anantha is a national leader in this consortium. He is a lot of you know this. He is a soft-spoken person who carries a very big verbal stick. And he has had an outsized influence and an outsized, very positive influence on this network, and turning this network into a true network for translational medicine that can really deliver in an exponentially improved way for patients. Another program in the clinical arena is the Rare Disease Clinical Research Network. One of these is partially based in Indiana. This one on mucociliary clearance. There's actually a new program which just got funded. We haven't updated the slides yet. That's also based in Indiana. But what these are actually 22 consortia of academic medical centers. Each one of them makes up multiple academic medical centers, focused not on a single rare disease, but actually on groups of rare diseases, which are grouped by some kind of commonality, either a common pathway like urea cycle, a common organelle like a mitochondrion or a lysosome, a common organ like a vasculature, and so looking at commonalities across disease. What's common about diseases that we can kill, hit two more birds with one stone. The other thing which is really innovative about this initiative is that it requires the involvement of a patient advocacy group as a research partner in the collaborative, not as frosting, not as just coming to the meeting to take pictures in, but actually being a partner in the work that the consortium does. And this has really been transformational for the productivity of these organizations. And I'm just going to run through now a couple of other public-private work that we don't have time to go through everything we do, of course. So I'm just going to give you a few examples of the way we're thinking about solving some of these problems, particularly focused on novel partnership models. So this is a program some of you may have heard about. This Discovering New Therapeutic Uses program. The problem here is addressed is that, of course, 80% of compounds or north of that enter the clinic never get approved. They either fail for efficacy usually, but also sometimes for tox, or they get deprioritized for business reasons. So what this suggests is that for every drug approved, there's 4 or 5 or maybe even more drugs, which the pharmas have put a huge amount of work into, but are non-performing assets, if you want to think about it that way. And from a patient standpoint, they are potentially new therapeutics that again, to go back to my football analogy, if the ball's sitting on the 10-yard line, could a new team come in and run a few plays and actually score for a different indication? And so here's the opportunity we just talked about. And so what happened was that NCATS served as a matchmaker. NCATS provided an informatics platform, sort of a match.com, that allowed the pharmaceutical assets to be listed, and then academic investigators who had ideas for diseases to which compounds with those targets might be appropriately indicated to apply and partner with the pharma to develop this for this new indication. So we provided the Match.com informatics and the review and the money. The pharmaceutical company, of course, provided all the drug, the API, the placebo, the compounds, project management, data, et cetera. And the academic investigators provided the idea and the patients. And these are the programs, and you can see the diseases on the left here, and I just want to point out that every one of these diseases is either a very poorly treated disease or is not treatable at all. 2 rare diseases and 7 common diseases. And I want to point out that one of them, true to form, is in Indiana. This is a program with Alan Breyer, who's in the psychiatry department with Anant at IU. This is a program, it's a Lilly compound. It's an estrogen receptor modulator, which is being tested for utility in schizophrenia. So this is a Lilly-IU collaboration that's funded by NCATS. Moving back in the preclinical world, so this is the usual chevron diagram you're all familiar with, and the way the operation works here is that most of the collaborators in this space are academic investigators who get stuck with it. They have a target, they have maybe they've done a little screening, they have a poor compound that they need to develop, they have a preclinical asset that they think might be useful for a disease, but they really don't have the expertise or the facilities to advance this. And so instead of the usual model, which would be that the poor academic investigator has to go learn all of this field and try to be a drug developer, which doesn't make any sense, or for a drug developer to become an expert in the disease, which also doesn't make any sense, we thought, well, we'll just put a joint project team together, and the people who apply to this don't get money. They get put on a joint project team with NCATS, so the academic investigator brings the disease target expertise, NCATS brings the translational expertise, and they go into one or more of these programs, depending on where they're stuck. And then out come the bottom come deliverables, and it's data and a physical substance. So it's an siRNA, it's a probe, it's a lead, it's a repurposed drug, whatever it might be. But each of these programs is also a dual-use project. We like them to have us move an individual project down the translational spectrum for an unmet medical need, or de-risk an individual target, or drug a new target. But they also need to tell us something general about how to do the process better. And I'll just give you a couple of examples. So this is where the staff comes from. We recruited virtually everybody at this part of the organization from pharma and biotech for reasons which you can imagine. But in order to get hired, you have to tell me not only what the state of the art is at the best places in the country, in the world, but you have to tell me how to do it better. Because the current state of the art, I don't think anybody thinks is optimal. And because we don't have the short-term commercial imperative that all of us had when we were in these environments, we are able to do the kinds of experimentation on the process that everybody would like to do, but you just can't do if you're on a return on investment environment. But we have the same expertise. And here's just one example, and it's a random example. It's not an Indiana example, but I thought I like this example. It happens to be on inhibitors of an enzyme called 12-lipoxygenase. It was discovered about 30 years ago. Nobody really understood what it did because there weren't pharmacological inhibitors. So there were SI experiments and knockout mice. I can tell you what a knockout mouse is if you're interested. But they're crude tools at the gene level or the mRNA level to figure out what these proteins do. And the field was really held up by the lack of inhibitors. So this guy named Ted Holman, who's an academic investigator at UC Santa Cruz, came to us, a biochemist, would know a drug if it hit him in the head. But our guys wouldn't know how to spell lipoxygenase, so that's fine. They formed a joint project team. Ted brings the LO experience. Our folks bring the high-throughput screening, assay development, med chem, chemical informatics, all that stuff experience. In about 2 years, we developed really nice inhibitors of this enzyme. And then what happened was really cool. That with this enzyme, we were able to, Ted and we were able to figure out what this enzyme actually does in living systems. And it actually turns out that it has really important effects in both platelet aggregation and in the autoimmune phenomena early in type 1 diabetes. And so we have a number of collaborations developing for those indications, and there's a collaboration with, so now an agreement with Sanofi to carry these projects forward. So it's a classic example of the kind of thing that NCATS does. We also have put a lot of work into repurposing for reasons that you can imagine. About 5 years ago, I said, well, gosh, everybody knows the Viagra story and all these other repurposing stories. But how do we systematize this? How do we take it from being happenstantial and serendipitous? How do we systematize that serendipity? And what was clear was what we needed was a complete non-redundant list of every drug ever approved for human use worldwide, whether it was generic, still on patent, it would have been on the market and then pulled off the market for a variety of reasons. And what was amazing to us back in 2006 when we started this is that nobody had ever done this. It was amazing. There was not a complete non-redundant list of every drug ever approved for human use. And you got to say, why? It's amazing. Nobody had ever done. Well, nobody had. It's tragedy of the commons problem. Nobody had ever had the mission to create such a thing. So it actually took us 5 years to do. And if you want to know why, it's all in this paper here. It sort of reads like a Stephen King novel, but it tells you why it took us so long. But all of this is now available. Informatics-wise, the whole database is on our website. We use this in collaborations. And one collaboration that I just want to highlight here is when we published this, the folks at Lilly, Alan Palkowitz and others, came to us and they said, we think this is so cool. What we want to do is pair this drug collection with RPD squared, our phenotypic drug discovery open innovation platform, to run every drug ever approved for human use across all of these really interesting phenotypic assays. And you guys do the drug husbandry part, and we'll do the screening part. We'll do the analysis together and put it all in a public database for everybody to use. And we're doing that. It's an incredibly important enabling initiative that's reaching fruition now. And just one example of what we put to, it doesn't really matter what the disease is, it happens to be CLL, chronic lymphocytic leukemia. In this case, we use this because patients with blood cancers have lots of cells that they're willing to give to you, so we were able to get about 1 billion cells from these CLL patients and from normal B-cell donors. And what we were looking for is could we find a currently approved drug which could kill the CLL cells, refractory CLL cells, but not the normal B cells. And it turned out that one of them was an old rheumatoid arthritis drug called Oranafin, which we never would have looked for because it's a rheumatoid arthritis drug. And then what we did was we very rapidly put together a collaboration that Deborah Lappin has been involved with from the beginning, who's involved in this meeting as well. It's a three-way collaboration between ourselves and nonprofit, the Leukemia Lymphoma Society, which is the largest funder of blood cancers, nonprofit funder of blood cancers in the world, and the University of Kansas that had a lot of expertise in clinical pharmacology. And by putting this collaborative together, focusing on the patient first, what would the patient want? Get into patients as rapidly as possible. We were able to go from that discovery of Oranafin to being in patients in a year. And that's the kind of thing that's really possible, but it requires a team. Another program which you might have heard about is this Therapeutics for Rare and Neglected Diseases program. It's the same idea as the lipoxygenase story I told you, except in this case, these are leads which need to be developed into proof of concept in humans for a rare disease, de-risk them enough that a private sector company would adopt them. And I just want to tell you one story here because it also involves an Indiana company, which is Lilly. So this is a really interesting story. So Lilly developed this drug, which is it's a PTH, parathyroid hormone analog for osteoporosis, and it actually failed for osteoporosis. But of course, for primary or secondary hypoparathyroidism, which is a rare disease, it might be, of course, incredibly helpful. But they had no business case. They couldn't make a business case to develop this internally, so they came to us and they said, "You want to do this together?" And we said, "Great, this is fantastic." So we're actually doing at our place. We're doing the tox and CMC works a little bit different for this indication as opposed to osteoporosis. And then if this reaches the point of proof of principle in a phase 2A trial, then Lilly has the option to reacquire it if they want to, or it can be licensed out to a third party for development of this indication. But it's another great example of taking what in this case is a compound that was developed by a pharma and moving it into development for a rare disease. Of the other programs, I would say about half of them were academics and half of them are small companies, a few other large companies doing these kinds of disorders. Okay, so lastly, toxicity. So this is the I just want to give you an example of one of the sort of re-engineering initiatives that we're doing that's independent of a disease. So toxicity, as you all know, is a major reason for drug failure. Still, it's about a third of drug failures preclinically and clinically. And so here's a really classic NCATS approach to this problem. Classic in the sense that it is meant to be transformational. This is not arithmetic, incremental change. This is aiming for transformational logarithmic change. Secondly, it's based on really cool cutting-edge technology, because we are unabashed techno geeks at our place. I don't mind saying that. And thirdly, it's a novel collaboration. So this is a collaboration between NCATS and DARPA, which is the Defense Advanced Research Projects Agency, the folks who brought you GPS and internet and all that stuff. And the goal here is to develop a microfluidic system by which human organoids could be cultured for up to a month in a modular system, so that potential therapeutics could be infused in artificial blood, go to one of these organs, and manifest an efficacy or a toxicity response, which is characteristic of a human organ. And then eventually to knit all these together to sort of create a little homunculus in a microfluidic system, if you will. So this is a program that started about 3 years ago. DARPA is doing a lot of the engineering, the microfluidic engineering, and NIH is doing a lot of the biology. Most of this is based on iPS cell technology, as you might imagine, but these are the organs which are being looked at here. There's about 10 organs, and each of the performers, as you probably know, DARPA likes to refer to these people, I really like that term, has to develop an architecturally and physiologically relevant system which models a human organ, but it's in a modular platform where in stage 2, which we're just entering, those modules can be linked to each other, sort of in a Lego bricks kind of way. And so there's a lot of very active project management in this program from the beginning. Now, let me say, before I go to what actually happened, I must say, when this project first came up 3 years ago, as you might have gathered so far, I am not one to shy away from big challenges, but I thought these guys were nuts. I thought this was never going to work. I mean, I thought it was wonderful. You guys are, you can do some great work, but you got to be kidding me. I was wrong. And I'm glad to say I was wrong. And I was wrong because I underestimated the power of convergence. And this is a convergence of IPS technology, tissue printing technology, biosensor technology, and microfluidic technology, so that all of these programs are actually ahead of their milestones and delivering in ways that far above actually we anticipated. And so how do you actually do this? It's an interesting sort of thing, just to run through this briefly. So what happens is you start with a scaffold, like sort of a matrix or something. Then you have to add cells, which are either primary cells or IPS-derived cells. They have to have a certain structure. If it's a kidney or a liver or whatever it might be, it has to have that proper structure. Sometimes you have to induce that structure. Sometimes the cells take it on themselves. They have to have the right spatial and temporal patterning. One of the things that we found is that it is only with spatial and temporal patterning, and actually often with physical perturbation that is normally seen, say in a lung or a heart, of the sort of physical stretching and unstretching, that the cells actually differentiate and do what they're supposed to do and mimic the normal adult system. These things sometimes have to be perfused. They have to be in bioreactors that keep them alive for a while. They sometimes have to be innervated by nerves, depending on what you're looking at, particularly with toxicity. Sometimes there has to be a host response component. There has to be a functional readout, how you're going to measure what the compounds do. And then there has to be a computational system to gather all the data. And this is just one example. These are cardiac muscular thin films. This is from work from Kit Parker at the Wyss Institute in Boston. And what you're showing is these are cardiac myocytes that, of course, you've probably all seen these now. If you put them in a petri dish, they will beat spontaneously. But it's been pointed out by many people that spontaneous beating of a heart muscle is bad. That is, if your heart muscles beat independently of each other spontaneously, you'd be dead. That's what's called ventricular fibrillation, right? They have to act together in an organized way in order to pump the blood. But they don't do that because they're immature cardiomyocytes. They only mature when you put them in this structure and make them stretch and unstretch with these little hammers here that are going back and forth. And when you do that, the cells differentiate and actually show appropriate responses to cardiotoxins. And eventually, the goal here is to create a body on a chip where one would have, for instance, you would be able to infuse a drug, go into artificial intestine, get absorbed or not, go to artificial liver, get metabolized or not, artificial kidneys, show tubular necrosis or not, or whatever one's looking for. And one would be able to have a variety of readouts like you see on the left, see on the right, sorry, and be able to correlate these, of course, with in vivo outcomes on the left. And the really interesting thing about this, despite the promise of the individual chips, is that, of course, if these are made from iPS cells, it should be possible to make individual person chips so that one might be able to look at variation in efficacy or toxicity responses in a microfluidic platform before you go into a clinical trial. Okay, so take-home messages. I hope I've convinced you that the opportunities in this space and the needs in this space are absolutely huge, and they are systemic. They're not one-off issues. They're systematic issues, and they need to be approached in a systematic way for systematic solutions. Secondly, the scale of the problem is such, the scale of the current lack of efficacy, speed, and cost is such that I do not believe that going after arithmetic incremental improvements is going to solve the problem. I mean, if you take about the drug numbers that we all know, right? 15 years, 99% failure rate, $2 to $10 billion, depending on how you do math. Okay, so 10% improvement, instead of 15 years, it takes 14 years. Instead of 10 billion, it costs 9 billion. You know, it's not appropriate to the task. This is like the Genome Project when it started. You know, we've got to improve by orders of magnitude, and I think it requires us to put our brains in a little bit different of a set. When you put your brain in that set where you realize you got to look for logarithmic change, the kind of things you think about is quite different. I think much more exciting, but quite different. So 21st century problems not going to be solved with 20th century structures. And lastly, the research ecosystem, and you're a great example of this, and I put NCATS right smack dab in the middle of this, are experimenting. Everything at NCATS is an experiment. I say this all the time. Everything from the programs we do to the science we do to the structure we have, everything is an experiment. And you're experimenting now here. We're experimenting in this process of just beginning to transform ourselves into the organizations and programs that are going to meet these opportunities and needs for the benefit of patients. And I was really delighted to hear the folks who introduced say, you know, this really is all about the patients, and you know, depending on where you go, that's not always the case. You know, there's sometimes lip service given to that, but I must say, whenever we have a problem in NCATS and somebody says we have a hard decision, what I ask people is to say, well, if you're a patient with this disorder, what would you want? Would you want people to share the data? Would you want people to act in a collaborative way? Would you want to? Then the answers are actually quite clear, and I think if we have that focus. And we have this collaborative teamwork notion that you all are implementing with such vigor here. I think the future is extraordinarily bright. So thank you for having me. Glad to take any questions.
Questions for Dr. Austin? Please just step up again to one of the microphones in the aisle. That was fabulous.
Thanks,
Dr.
Dan.
Lecklider.
John Lecklider from Lilly.
Ah.
Chris, you've made great progress since that day we
Yes.
sat in my office a few years
Yes.
ago, and sort of you asked, "What hath God wrought?"
Yeah.
And
You gave me my marching orders.
well, you know, you had a slide, and I didn't notice or not one of the circle slides whether you connected to FDA.
Yeah.
I trust that was on there.
Yeah.
Can you talk just a little bit about what your center is doing to reach out to FDA? Some of these things will require a change in mindset on the part of the regulators.
Yeah, it's a fantastic question. And yeah, one of the first people that I called when I got this job was Peggy, actually. And actually, I'm having to step out of this meeting actually at 11 o'clock to do a search for one of their big people there. I think a lot of what we're trying to do together is induce scientifically based culture change in that organization. There are some fantastic people at FDA, and you know them. But it is an organization like the organization that I run and the organizations that you run that has an aversion to risk, and humans are like that. And the only way that we're going to be able to fix this, I think, is to get people the data that they need, the reviewers, et cetera, the data that they need to feel comfortable that they're not taking a risk. And what I find is that we are in a really unique position at NCATS, and NIH in general, but in NCATS in particular, because of the fact that about half of our 300 people came from pharma and biotech. You know, we speak this language, and so we are able to work with them on individual projects or on larger projects, showing data in a very free way, which really is very beneficial to them, where they're able to show their dirty laundry, we're able to show our dirty laundry, we're on the same email server, and we're on a first-name basis with all these folks, and it's really leading to a level of trust, which we're hoping, and this is really the goal, that this will be viral change within the organization. And I'm actually very hopeful that this is going to work. I think it's one of the really special things that NCATS can do.
Michelle McMurray-Heath of Faegre BD Consulting. I'm over here, Chris. And I actually just spent 4 years at the Center for Devices, and I can vouch for what Chris just said. He really has the focus of FDA, and people are really looking to his leadership to see how we might have new paradigms for judging these applications of very innovative approaches. So he's absolutely right. He has a huge impact within FDA, and I hope he continues to. It was actually not my question. My question was, I was so intrigued by your slide about T1, 2, 3. People tend to think translational research is one thing, and you get a drug approved or a device approved, and that's the end of the conversation. But this whole trajectory is so fascinating. What are you doing within NCATS to partner with new clinical trial networks like PCORnet to take advantage of getting those real sea changes in how healthcare is provided as these innovations come online?
Yeah, it's another great question. I've had a few people ask me, said, I understand all this drug development stuff, it's great, but in fact, we know that for many disorders, 50% of people who are prescribed a medicine never fill it, and the 50% of those who do fill it never renew it. So if we know that 75% of people who are prescribed a current drug never take it, why are you trying to make new drugs? Why don't you focus on the fact that adherence is bad? Because you're going to have a much bigger effect there. Now, of course, it's a portfolio issue, right? We don't want to do one or the other, but this is really something which NCATS is very much focused on, and so what we're trying to figure out is how, just as you said, how do we position ourselves vis-a-vis the other organizations, like it's not only PCORI, but it's HRSA and ARC and all those other organizations. We actually have a very close relationship now with PCORI, such that Petra Kaufman is our head of clinical, and Joe Selby have regular phone calls. We have a joint working group. We're writing a paper together, trying to figure out inducing our respective networks, the CTSA network that's more focused on development of new interventions and sort of early clinical studies, and PCORnet, which is more on the distal side, to say if you're a patient, it shouldn't matter, right? So you shouldn't have to sign up for two different databases, right? And we ought to be able to take data from PCORnet and compare it with what goes on the CTSAs and vice versa. It ought to be missable. And so that is, what's interesting is when we first got into this about 6 months ago, it was presented to us as a technical problem. Can't do it. IT systems are incompatible. It turns out that's the cat ate my homework answer. It's not a technical problem. It's actually a cultural problem. And once the leadership of NCATS and PCORI said, no, we're playing from the same playbook for the benefit of patients, then all the players said, oh, okay, so we'll now play together. And I think the good thing about this is, I guess the way to put it is it reminds me of the situation in genetics about 20 years ago, when people realized that these problems are so huge that they can't do it alone. And they've tried, and it doesn't work alone. And so PCORI doesn't want to do it alone. We don't want to do it alone.
That's fantastic. Great, thanks.
Okay, we have one. time for one more quick question.
Chris, Dr. Collins notes that it takes about 14 years and $2 billion to bring a new drug to market, factoring the failures. And you've noticed some other estimates are higher. Dr. Collins also says that at a recent roundtable on the 21st Century Cures Initiative, one of the barriers that we face is intellectual property on the repurposing drugs where the patent term is down. We've been partnering with patient groups and others to try to come up with a system where the intellectual property clock would be restarted at FDA approval, so we can try to correct that mismatch where the current patent law only allows up to 5 years to be put on, but the development time is so long. What do you think about proposals in that area?
There's a proposal, actually legislation, which has been introduced twice called the Modern Cures Act, which addresses this issue of dormant therapies, and it's essentially it's a reanimation of dormant therapies, a reapplication to a period of exclusivity for an off-patent drug which has a new indication which otherwise wouldn't be developed. And we run into these problems all the time. That drug collection that we put together, it's about 2,800 approved drugs worldwide, and about 90% of them are generic. And so when we do a screen, 90% of the time we pull up generics, and they're really hard to develop through clinical studies. So I think there's a lot of promise in that. Interestingly, there is it's run into not a scientific argument, but actually a political argument in the Congress. There's such a focus, and understandably so, on drug prices. Anything which would increase a drug price is a lot of people just react kind of in a reflex way negatively. We've just actually been asked, just yesterday actually, I got a phone call from some folks asking if we would be willing to just opine on the science of this. What are the possibilities? Don't take a policy position. We can't take a policy. We don't want to take a policy position. But just from a scientific point of view, is there a real scientific opportunity here? And what is the medical problem? What is the problem in the medical opportunity? So I think there's a lot of room to run here if we can get that problem solved.
Fabulous.
Good. Yeah, thank you.
Please help me thanking Dr. Christopher Austin again. What a fantastic talk. I learned a ton. I always learn a lot in these sessions, but I really learned a lot in that one, and I really appreciate it. I learned that you can fit Dr. Francis Collins' work in a toaster. And I learned that translational scientists like football. I was really glad to learn those things. In all seriousness, there were a lot of really important messages there that I think we all appreciate and feel very positive about looking forward, especially just kind of the systems thought on organizations like yours and like FDA and others, that we need 21st century approaches to those types of opportunities and challenges that are out there. And it's a team sport. I thought that was a great analogy there. As well. And so really appreciate your comments and hope you can stay throughout as much of the day as you can.